Hello, I'm by the name of Emmanuel Ategekimana. I'm a student of Nestle Engeri, Faculty of Biomedical Laboratory Science. I'm a student of year 3. I'm going to present the applied immunology. Here are the overall of the question I'm going to present. It's like a 5 question from 6 to 10. First question I'm going to present is about the central tolerance, how it can prevent autoimmune disease, what is that autoimmune disease, and 12 medical important autoimmune disease, their cause and clinical manifestation. The second question was about the checkpoint immune and why they are important. The third is how cytokines regulate immune response, and the fourth one is about the monoclonal antibody production and how they work in immunotherapy. And the last three is how the principle behind the checkpoint inhibitor. Let me start on the first question, central tolerance, how it can prevent autoimmune. First, central tolerance is the process of eliminating self-reactive immune cells during their development in primary lymphoid. If I say primary lymphoid, it's like it's thymus and bone marrow. If I say thymus, it's where T cell will be matured, and bone marrow is where a B cell will be matured. As you have seen on this slide, in T cell in the thymus, mature T cell will undergo negative selection where those strongly binding to self antigen, where on the B cell it is on the bone marrow where self-reactive B cells are either directed under receptor or editing to modify their specificity or become an energy. So on the autoimmune disease, the immune disease is like the malfunction of immune system, how our immune system mistakenly attack its own cell or a tissue. On this diagram on the right, it's where the B cell attack their own cell produced in the bone marrow, and T cell, like T cytotoxic, instead of infecting the targeted cell like cancer cell, they destroy their own antigen against on antigen. On the first disease is rheumatoid arthritis, where it's caused by genetic predisposition and other things. And on the clinical manifestation is it can cause joint pain, swelling, stiffness, systematic inflammation, and other. Go on the second disease, which is type 1 diabetes mellitus. Is caused by like autoimmune destruction of the pancreas B cell. Well, if I say pancreatic B cell is where insulin are produced. If the body mistaken attack the B cell, it means the level of insulin will decrease and cause the type 1 diabetes. This one will cause the high level of sugar in the blood, which is called the hyperglycemia, cause insulin help sugar or glucose to enter into the cell. If it didn't enter, they will be high in the blood. That's hyperglycemia. Polyuria, weight loss, and predyspea, which is increase in thirst, and those polyuria to urinate more urine. The third one is systematic lupus erythematosus, where it is caused by autoantibody. Autoantibody is where our own antibody are being attacked. This can cause rash, arthritis, and nephritis. Go on this fourth one, which is multiple sclerosis. It's caused by immune attack on central nervous system, myelin, also genetic predisposition. It can cause this multiple sclerosis. On the clinical manifestation, it can end up with muscle weakness, vision loss, coordination problem, and other. And the fifth one is Graves' disease, where it's caused by autoantibody stimulating TSH receptor. TSH is thyroid stimulating hormone that is secreted by thyroid gland. This can ending with hypothyroidism, which is increased production of thyroid hormone and weight loss is one of the clinical manifestation. Also, we have Hashimoto thyroiditis. Hashimoto thyroid TTC is caused by autoimmune destruction of thyroid cell. When the body mistakenly take the thyroid cell as a foreign cell and mistakenly attack it, they cause what we call those Hashimoto. On the clinical manifestation, it can ending with hypothyroidism, fatigue, weight gain, and cold intolerance. Also, cirrhosis is one of the disease of immune system. This can be caused by T-cell mediated skin inflammation. On the clinical manifestation, I think scalp plaque and itching. Have ankylosing spondylitis, which is caused by HLA-B27 association, and ending with chronic back pain, spinal stiffness, rheumatism. On the ninth disease is celiac disease, which is caused by gluten-induced autoimmune endotherapy. On the clinical manifestation, it can end with diarrhea, bloating, malabsorption, and weight loss. The 10th disease is myasthenia gravis, which is caused by autoantibodies against the acetylcholine receptor. And on the clinical manifestation is where we have a muscle weakness, fatigue, and dropping eyelid. Also, we have pernicious anemia, which is caused by autoantibodies against intrinsic factor. Those factors which can cause the pernicious anemia. Or we can have a B12 deficiency, fatigue, and gastritis. The last three we have inflammatory bowel disease, which is caused by Crohn's disease or ulcerative colitis. The clinical manifestation, we have abdominal pain, diarrhea, weight loss, and blood in stool. That is good. The second question we have on the immune checkpoint and why they are important. Immune checkpoint, firstly, are those molecules found on the immune cell that regulate the immune response just to maintain self-tolerance and preventing the tissue damage. It's like to block the pathway. They help to maintain self-tolerance and avoid autoimmune damage. Have example which is cytotoxic T lymphocyte antigen 4, which can inhibit the T cell activation by competing with those CD28 for B7 binding on antigen presenting cell. Also, we have programmed cell 1, programmed cell D1, which also bound to programmed cell D1 or programmed cell D2. Also, this will inhibit T cell activity by promoting immune tolerance. They tolerate. If they tolerate, they can attack only foreign antigen instead of attacking their own antibody. They are important by preventing autoimmune by suppressing overreactive of T cell. This immune checkpoint can prevent our T cell to react over, or to act as over and start to take their own cell as a foreign. Also can this immune checkpoint limit tissue damage during the infection or inflammation by competing those other molecules for binding to B7 like this toxic T lymphocyte antigen 4. Also, thermal exploratory checkpoint like PD-L1 and PD-L1 to evade immune response, making this pathway key therapeutic. As you have seen on this diagram, we have here immune checkpoint, you have thermal cell or antigen presenting cell, you have T cell, you have molecule, or we have cytotoxic T lymphocyte, which is responsible for destroying the infected cell or cancer cell. So you have this programmed cell death, and you have this one and this one, this programmed cell death and the antigen and this cytotoxic T lymphocyte, they compete with those molecules to bind to B7 and the immune system. Here we have B cell. We have also T helper cell. This T helper cell or T helper cell too, they can secrete what we call interleukin. This interleukin is a molecule of cytokine where they can enhance the immune response and help B cell to produce memory cell and plasma cell, where memory cell provide long-term immunity, and the B cell and this plasma cell, they can produce antibody. Also, as you have seen on T cell, that is on the B cell, and T cell have infected the cell, T cytotoxic. Just we know T cytotoxic and the natural killer cell, they work together with this to destroy those infected cells and work on this mechanism of action, how they work and how they work in immunotherapy. We have first one, targeted binding. On the targeted binding, monoclonal antibody just target the specific antigen that those marking cell for destruction. Those monoclonal antibodies just target the specific antigen. They didn't destroy the specific one. The second one, have immune activation. Those monoclonal antibodies engage immune cell like natural killer cell and macrophage, which is used for engulfment, and phagocytosis, natural killer cell for destroying the infected cell. Also, you have through Fc receptor to induce antibody-dependent cellular cytotoxic as one of the important of antibody. Fc is a fragment crystallizable. Now on the third one, you have complementary activation. On complementary activation, those monoclonal antibodies, they trigger the complementary cascade, tries the targeted cell. This one can help for lysing the cell to destroy completely the cell. On the last one, they block signal. They do as like those checkpoint immune. They inhibit the signaling pathway essential for thermal growth or pathogen survival. They block all the immunotherapy. I have the reference that can help me to do this work. Thank you for watching this video. I highly appreciate it.